All right, so let's start. Hi, everyone, and welcome to SCIEX webinar, PFAS in the pharmaceutical industry. My name is Mandana Fas, Senior Manager, Pharma and Biopharma Market Development EMEA, and I am excited to be your host for today's session. Before we begin, I would like to briefly explain the webinar console slides and review a few housekeeping items. And now into housekeeping items, please. All attendee microphones will remain muted throughout the webinar. If you need assistance during the session, please use the chat box to contact the webinar team. We encourage you to submit your questions at any time using the QA panel. We will address as many questions as possible during the live QA session at the end of the presentation. If we are unable to answer your question during today's webinar, a member of our team will follow up afterward. This webinar is being recorded and will be shared with all registrants following the event. Please note that the QA discussion will not be included in the recording. So before we move to today's presentation, I would like to highlight a few additional resources available to you. The slide includes a link to our pharma and biopharma landing page, as well as 2 technical notes covering PFAS analysis in pharmaceutical active ingredients and pharmaceutical packaging materials. If you would like to explore the topic in the more depth after today's webinar, I encourage you to access these resources. And now into speaker's bio. And now it's my pleasure to introduce today's speaker, Dr. Cathy Lane. Cathy Lane joined SCIEX in 2006 as a support specialist, and in 2021 took up the role of EMEA applications lead for pharma and peptide quant. She has more than 20 years experience in the development of LC-MS methods across multiple application areas. She obtained her PhD in mass spectrometry-based proteomics from the University of London School of Pharmacy in 2004. With that, I will hand the presentation over to my colleague, Cathy. Welcome.
Thank you, Mandana. Okay, so let's start with an agenda. I'm going to begin with an introduction to PFAS and then move on to talk a little bit about PFAS in the pharmaceutical industry. We'll cover some analytical challenges when monitoring PFAS by LC-MS, and then I'd like to talk you through the 2 case studies that you saw in the tech notes that Mandana shared with you earlier. So we'll look at a sensitive method for the quantification of PFAS in a fluoropharmaceutical API, and then we'll look at a sensitive method for the quantitation of PFAS in pharmaceutical packaging containers. PFAS are a large family of synthetic chemicals that have been widely used in the past 80 years in a vast range of applications, spanning consumer, commercial, medical, and industrial products. They're well known for their resistance to heat, water, and oil, which means they have multiple uses, for example, in non-stick cookware and waterproof clothing. They're commonly known as forever chemicals because they break down only very slowly over time, and this can cause long-term contamination issues. In addition, some PFAS are known to cause harmful health effects in humans and animals, such as cancer and developmental issues. Regulatory agencies, such as the EPA and the ECHA, have published multiple regulations to control the use of PFAS and to monitor their presence in consumer products and the environment. PFAS have multiple uses in the pharmaceutical industry, including as surfactants and emulsifiers in drug production, in packaging because of their non-stick nature, and also in medical devices such as implants because of their chemical inertness, durability, and biocompatibility. To ensure the safe production of medicines, analytical testing labs and pharmaceutical companies must meet regulatory thresholds, standards, and testing methods as they evolve, and they are evolving quickly. The PFAS used in medical devices, such as PTFE, are not currently restricted, but the FDA is continuing to monitor their safety, and the ECHA has published a draft proposal to restrict PFAS in 15 sectors, including medical devices, packaging materials, and PFAS manufacturing. The ECHA aims to complete its scientific evaluation of the proposed EU-wide restriction of PFAS by the end of this year. And I've put some links there to various ECHA websites and some responses to the ECHA position on PFAS. As this landscape evolves, increasing regulation will necessitate effective, sensitive, and robust analytical methods for PFAS measurement. Moving on to the analysis of PFAS, there are multiple challenges associated with measuring these compounds by LC-MS. PFAS compounds vary widely in terms of their structure and their physical characteristics, and this presents challenges for the optimization of the analysis of multiple PFAS compounds in a single method. PFAS are everywhere, so background contamination is always a potential issue. Good baseline separation and chromatographic conditions are needed to ensure proper and accurate quantification of PFAS. And currently, there are no published regulations on PFAS threshold requirements in the pharmaceutical industry, which of course presents challenges in itself because you don't know how sensitive your method needs to be. Presumably for now, the more sensitive, the better. Right, let's move on to our case studies. So I'm going to show you data first on a sensitive method for the quantitation of PFAS in a fluoropharmaceutical API. On this slide, you can see sample preparation details and also LC conditions. So we have a working stock solution containing 23 PFAS plus internal standards prepared in a methanol water mix and serially diluted to create calibration line standards in a concentration range of a picogram per mil up to 20 nanograms per mil. Our test API was prepared at 2 mg per mil in the same solvent, and we also prepared spiked samples containing 0.05 nanogram per mil of our PFAS compounds in 2 mg per mil of the API. The LC conditions are shown in the lower half of the slide. So we're using a delay column. So delay columns are commonly used in PFAS analysis to separate any PFAS that might be contained in your mobile phase or in the LC system from the PFAS in your sample. So in this case, we're using a delay column from Phenomenex, a Gemini column 3 by 50 millimeters. Our analytical column is a kinetics phenyl hexyl column 3 by 100 millimeters, temperature of 40 degrees, and a flow rate of 300 microliters a minute. Mobile phases were 5 millimolar ammonium acetate and water and 5 millimolar ammonium acetate and methanol with an injection volume of 2 microliters. You can see the gradient in the table on the right-hand side of the slide. And one thing to note is that we're using the diverter valve to switch to waste at 10 minutes. And this is to divert the API itself, which is present at a very high concentration, to waste away from the mass spectrometer after our final PFAS compound has eluted. And so in order to monitor the API, we're using UV detection at 220 nanometers. Here you can see our mass spec method parameters. So samples will run on a SCIEX 7500 plus system. You can see the MRMs used to monitor the PFAS and their internal standards in the table there. And then source and gas parameters are on the right. So obviously when you're optimizing source and gas parameters, you're having to use a kind of compromise to get the best sensitivity as possible for a wide range of PFAS compounds. Here you can see chromatographic separation of our PFAS compounds and our API. So in the top panel, panel A, we've got representative extractine chromatograms of our PFAS, and these are at 0.05 nanogram per mil in 2 mg per mil API. And in the lower panel, you can see the UV trace, which shows the elution of our API. And you can see that we have clear baseline separation there between the PFAS compounds and our API. And this slide compares our PFAS in solvent with PFAS in API. So the top trace A there, shows the overlaid total line chromatograms of our calibration curve sample at 0.05 nanogram per mil in solvent and our 0.05 nanogram per mil PFAS spike in 2 mg per mil API. And you can see that the peaks overlay very well, and this just indicates there are no significant matrix effects from the API that is affecting the detection of our PFAS compounds. In the lower slide there, we've got individual XICs of all the PFAS, both in solvents and in the API. And again, you can see that the responses are very similar between the two different solutions. Moving on to quantitative performance data. What I've shown in this slide is representative sensitivity data for 4 of our target PFAS compounds. So we've got PFOA, PFNA, PFHXS, and PFOS. And for each PFAS, we're looking at the blank, the extracted ion chromatogram at the LOQ, and the internal standard extracted ion chromatogram. So in this case, for these 4 PFAS, you can see that LOQs are between 0.002 and 0.005 nanogram per mil. And you can also see that we've got no contamination or interferences in the blank solvent samples. Looking at linearity, I've shown representative data here for 2 of our PFAS compounds. So PFHX in the top of the slide and PFOS in the lower part of the slide. So we've got linear dynamic range of at least 3.3 orders of magnitude with R-square values of 0.995 or above. On the right-hand side of the slide, you can see the table with accuracies, percent CVs, and you can see that our accuracies are within 12% of the nominal values, and we've got percent CVs of less than 9%. And in this case, each concentration level was run in triplicate, and you can see that from the calibration lines. Just to show you some data on how the sensitivity of our method compares to an application where there are published thresholds, what you're seeing in this table is a comparison of the LOQs for our 4 representative PFAS compounds with the CDC minimal risk levels for adults and children in drinking water. So we've got PFOA, PFNA, PFHXS, and PFOS with the LOQs from our method in the central column, along with their CVs and average accuracies at the LOQ. And then we've got the minimum risk levels in drinking water for adult and children with the equivalent amounts in PPT on the right-hand side. And what you can see for each of our compounds, we're at least 10-fold more sensitive than the MRL for adults and at least 2-fold more sensitive than the MRL for children. Now, this table is quite difficult to look at, but it gives us a lot of information. So it summarises the quantitative performance for all 23 of the PFAS compounds in our method. So you can see from the first column here that our LOQs range from 0.001 nanogram per mil for 8,2 FTS up to 0.02 nanograms per mil for PFHPA. We've got linearity over at least 3 orders of magnitude, linear dynamic range, with R-squared values with at least 0.993. Accuracies at LOQ are within 5% of the nominal values, and CVs are less than 9% throughout for triplicate analyses. Now, recovery was evaluated by comparing the analyte peak area of the PFAS calibration standard at 0.05 nanogram per mil with the peak in the spike in API. So testing in this case was performed using 6 replicates, and we found good assay recovery within 11% variability, and that's reflected in the TICs I showed you earlier where we overlaid the traces from the calibration curve standard with the traces from the API SCIEX. So let's have a look at the quantitation of PFAS in our API. So what you can see here is for our 4 representative PFAS, we've got our API blank on the left-hand side and our SCIEX on the right-hand side. And what you can see actually is that we didn't detect any PFAS compounds in the API, and this is true across the board for our PFAS compounds. Okay, so now I'm going to move on to case study number 2. So I'm going to show you data on a sensitive method for the quantitation of PFAS in pharmaceutical packaging containers. So in this case, we're using a working stock solution containing 34 PFAS compounds, and this was diluted to give us calibration curve samples in the concentration range 0.001 to 10 nanograms per ml. For our packaging container extraction, we used a 10 ml aliquot of methanol, and this was added to the pharmaceutical packaging container, which was an ophthalmic drug container, weight 3.85 grams, and you cut up the pharmaceutical packaging container and you soak it in the methanol. And then for our spiked sample, we have a container that's spiked with 0.01 nanogram per ml of PFAS in 10 ml of methanol. The samples were sonicated for an hour at 50 degrees, after which a small amount of water was added. Samples were transferred to autosampler vials for analysis. And this method follows a method from the EPA, basically. In the lower part of our slide, we've got our LC conditions. So slightly different setup this time than the setup that we used when we were looking at PFAS in an API. So this time we're using a Lunar Omega delay column and a Lunar Omega PSC18 analytical column, 50 degrees C, flow rate of 600 microliters a minute, and our mobile phases were 2 millimolar ammonium acetate in water and 0.05% acetic acid, and methanol with an injection volume of 3 microliters. And you can see our gradients on the right-hand side of the slide there in that table. Coming on to our MS method parameters, you can see the MRMs that we used for monitoring our PFAS compounds in the table. And on the right-hand side, we've got our source and gas parameters, which are necessarily going to be different actually to the previous method because we're flowing much faster this time. Okay, now let's have a look at our chromatographic separation of 34 PFAS compounds. So here you can see a representative extracted ion chromatogram of our PFAS compounds at 2 nanogram per ml in solvent. And you can see that we've got good separation across 34 of our PFAS compounds. Moving on to sensitivity. Here you can see 4 PFAS, looking at the blank solvent sample and then the LOQ samples. So we've got PFOSA, 8.2 FTS, Adona, and 4.2 FTS. And you can see that for each of these PFAS, we're looking at LOQs of 0.001 nanogram per ml. And we've got no background interference in our blank samples. And as you'll see later on, we actually achieved LOQs of less than 0.005 nanogram per ml for all 34 of our PFAS compounds in this method. Looking at some representative linearity data, here you can see data on 8.2 FTS and PFOSA. So in each case here, we've got 4 orders of magnitude linear dynamic range. And on the right-hand side, you can see quantitative statistics. So we've got assay accuracies within 10% of the nominal concentrations, and our CVs are all less than 9%. Moving on to look at the results from spiking the PFAS into our packaging container. Here you can see data on 4 representative PFAS compounds, so the same compounds that I showed you in an earlier slide. In each case, we've got the blank packaging container extract on the left and 0.1 nanogram per mil spike on the right. And again, you can see that the blanks are very clean, and we didn't detect any PFAS compounds in the pharmaceutical packaging container. So this slide shows quantitative performance and recovery evaluations for all 34 of our PFAS compounds. So again, not easy to look at, but there's a lot of information here. So our LOQs for 34 compounds range between 1 picogram per mil and 5 picogram per mil. We've got linearity of between 3.2 and 4 orders of magnitude, linear dynamic range, with R² values of at least 0.99. Our accuracies at the LOQ are within 9% of the nominal values, and our CVs are all less than 14% at the LOQ. Okay, so recovery again was evaluated by comparing the peak area of each PFAS in the 0.1 nanogram per mil calibration line standard with the area in the pharmaceutical packaging spike at 0.1 nanogram per mil. So in this case, recovery testing was performed using 3 replicates for all 34 of our PFAS compounds, and we had good assay recovery with an overall recovery of more than 96.7%. Okay, now finally a summary and some conclusions. I hope that I've shown you methods for the sensitive quantitation of PFAS in an API and in pharmaceutical packaging containers. We achieved low picogram per mil LOQs for all of our PFAS, and this was analysed on the SCIEX 7500 Plus system. We achieved good chromatographic separation between our PFAS compounds using a simple gradient system. And for our first case study, we showed baseline separation between the PFAS compounds and the API. We also had good assay recovery from an API and our pharmaceutical packaging container for all of our PFAS tested, but we didn't detect any PFAS compounds in the unspiked API test solution or our packaging container extract, which is a good thing.